All right, good afternoon. It's time for a little asynchronous lecture. We don't have to meet in lab this week, but I do want to give you the lecture that I would give you in lab if we were all meeting. And so this is going to have two parts. There's going to be one part that is more general, and there's going to be one part that is more specific to our project. First off, let's talk about siderocalins. We're going to talk about the history of what we know and where I got the project from. They were originally, we knew always that they were calyx proteins, that they had a cup, a chalice, a calyx, and that they bound something. And when you don't know what something is, you usually call it a lipid because usually it has some kind of complex carbon structure. So they call it a lipid chalice or lipocalin. Notice that this just means they didn't know what it bound. And so it had unknown function. We knew it's important in the immune system. And there's an interesting story about how they found out it bound iron, which means siderocalins became their name. So lipids are just anything that's hydrophobic. We're going to talk about that, but calin comes from calyx, meaning cup or chalice, and they're basically like the blue cup-colored protein here, and it binds a red ligand that fits inside the cup. It's very easy to evolve the inside of the cup so that it fits different things, different shapes, different charges. And so there's a different sized calyx region that will be for their different functions, and you can have diverse sides and insides of the cup. So basically, way back in 2000, before I was still in grad school, I worked as a postdoc with Roland Strong. And so Roland Strong at the Fred Hutch, he was my PI when I was a postdoc. Before I became a postdoc, he studied the structure of immune proteins, and his graduate student at the time, Dave Getz, and they published a structure of this new lipocalin they were interested in, and it started with an unknown function. So they gave it a really terrible name called NGAL. It's what you do in immunology. And they just knew it was important to immunity, and they were hoping that they could be able to maybe see what's inside the cup. But remember, X-ray crystals only give you the electron density, and they weren't even able to see electron density. They just had an empty cup. That's kind of what they expected. And so they sort of modeled what different molecules could be in there. They knew the charges, they knew kind of the shape, and so they compared it to other lipocalins that they knew what was in the cup. So like they knew that this other lipocalin bound retinoic acid. This other lipocalin binds a fatty acid called palmitate. This one binds heme. If you look very carefully, you can see it's a little square heme on the side. And so they said, we know that it's a very different charge and shape. We don't think it binds any of these things, but we wonder what it might bind, and they modeled something and that ended up being completely wrong, so I'm not going to tell you about it at all. They measured the width of the calyx, the size of the lid. They sort of modeled how different things could fit inside it, but just realize that this is tricky to do, and it's hard to prove, but you can at least get a start with the idea of, well, you can know it's not one of these other things, like retinoic acid. It doesn't appear to be that. And they had this cutaway view of the cup, which I think you might want to try to replicate sometime using the slab or clip function of PyMol, which gives a nice side view of how things fit in. Even the thing they were proposing would fit in didn't really fit in well, and they were kind of showing you that with this. The blue part doesn't fit into the red part very well. There's the retinoic acid. And so you can see that they could be pretty sure what it was not. They couldn't be sure what it was. Until one of those things came along in science. Dave made the protein, and usually you make them in BL21 cells, which are really good at expressing lots of protein. They're genetically just good for that. One day Dave expressed the protein in DH5 alpha cells, which are usually used for cloning DNA, not for expressing protein. And when he purified the protein, he saw that it was orange, and immediately he knew this is probably an iron-binding protein. This is our first sign that it was not just a lipocalin, but a siderocalin, an iron-binding cup. And this is what he shows, and so they have the paper where part of the evidence in the paper is they just show you, normally proteins don't absorb any color. Here they're absorbing wavelengths so that they look orange. And iron is orange. Iron is important. Iron would fit in the pocket. And the other thing is there's another piece to the puzzle, because bacteria want iron too. The pocket's way too big for just iron, and there's not much function in just binding iron. We've got other proteins that do that. But what this binds is it binds the bacterial iron binding molecule. So bacteria make organic molecules called siderophores. The siderophores will surround the iron, they will chelate it, they'll sort of be a claw around the iron, and then what they will do is they'll take the iron back to the bacteria. Well, we don't like bacteria, no offense, but they cause disease. We need an immune system to take care of them. So one way to interfere with bacterial growth would be to grab onto their grabbers. If they have the siderophores to grab iron, we make a protein that will grab their siderophores and keep them from returning to the bacteria. The bacteria don't have enough iron, they basically starve from that, our immune system functions. And so they followed that clue, and it took them a couple of years, but these are all the people that I worked with in the lab. This actually was published when I was a postdoc in the lab, so I knew all these people. One of the pieces of evidence in the paper was that you don't just look at it and say that looks orange. You actually are going to take the spectrum of it from the visible wavelength. You know it absorbs in the visible wavelength. It lets the reddish 700 nanometer light through, but that means it absorbs around 500 to block that light. And what they did is they took a spectrum of the protein. Remember, it was called NGAL, that's its old name. They took the spectrum without any ligand in it, and of course it's as clear like usual proteins are. But then they took the spectrum of the orange kind that you got from the DH5 alpha cells, and lo and behold, it has this big peak right around 500. We're hoping that you might be able to see the same kind of thing if you get an orange siderocalin. They also have other techniques, atomic absorption, which we can also do here. And they showed that iron was present, and not just present, it was present in a 1-to-1 ratio. That means 1 mole of calyx binds 1 mole of iron and 1 mole of siderophore, therefore. And so this is a really nice experiment that shows, this really showed that it was the iron. So the way the siderophore works, this is a particular common siderophore called enterobactin. Enterobactin has 6 oxygens that will chelate the iron in the middle, and this fits perfectly by size and charge into the siderocalin pocket. And so animals make siderocalins, bacteria make siderophores, and everyone's fighting over the iron. Now there are different siderophores, and what's cool about siderocalin is the bacteria can make lots of different siderophores, and siderocalin finds a way to bind them all. There's another one called carboxymycobactin, which is from mycobacteria, and if you look up here, it's a rather different structure, although if you look hard you can try to find these OHs. It has similar kinds of groups that will form a 6-membered claw. And then down here's enterobactin with 3 rings, 2 OHs each, that forms 6 OHs that will be the claw around the iron. And so you want to look at these. What we found out is that siderocalin actually binds both of these, and it also binds some of these other ones as well, which has some structural similarity, but that's one of the reasons why it's not necessarily a perfect fit for enterobactin, because siderocalin doesn't just bind enterobactin, it binds other things as well. And because they have different shapes, especially the carboxymycobactin, they will fit into it slightly differently and contact different residues. Different residues will be important for binding the different siderophores. And so how the siderocalin works is it has one big pocket that has sort of 3 little pockets inside it, sort of a triangle, and you can see it's negatively charged to bind all of these red oxygens that are on the siderophores. And then those are there because they have to bind the iron in the middle of things. And so they found the structure of human siderocalin bound to both enterobactin and to carboxymycobactin, which is CMB. You've already seen enterobactin in the structure that we had last week. It was ENT. And so these are the different ligands that you can find in the pocket. Somehow it's flexible enough to be able to bind all of these, and they even traced out the different amino acids that make contacts. And in enterobactin, the amino acids on the top are important for binding, including, look up here, pocket number 3 has lysine 125 right next to it. Lysine 125 is forming good interactions with one of the rings that is in the pocket right there. Lysine 125 also forms an interaction with carboxymycobactin, but it's to a different part of the molecule, and there are different amino acids that contact different parts of the molecule. Well, we know what all of these amino acids are, so one of our first questions is, when we give you a new siderocalin sequence from another species, it's going to have different amino acids. Are these important amino acids the same or different? The same, similar, or different? You kind of have three possible answers for each one of them. Because you know your amino acids, you can actually say, if you see an arginine in the place of the lysine 125, you can say it's similar, because they're both positively charged large residues. And so we have all of this in I want you to go through and I want you to look at these different residues. And I don't want you to look at them in the sequence. I want you to look at them in the aligned structure. You want to take the 1DFV structure, which is our basic structure that we have for this, and you want to see where those line up in space with the residues that are in the mammalian model that you're going to be given. So they went and once they did human siderocalin, lots of people did different related proteins from different species. Some of these aren't even siderocalins really, but all of these, I believe, bind some siderophores at the very least. Yeah, looking up and down, I see that I think all of these are siderophore-binding. And you can see that there are different species identities. If you look up and down this, you can see like 81 for mouse siderocalin and rat siderocalin are 81% identical. Human and rat are 62, human and mouse are 60. If you get down to 20, when you get down to 20, you are not quite sure about the structure anymore, so you need to do a little extra computer work. If you have 60% identity, you can be almost certain that these also bind siderophores. If they bind around 20, maybe they do, maybe they don't, or maybe there's a difference in their cup that will have them bind something different. If you look down here at this chicken XFABP, it's a weird name, but FABP stands for fatty acid binding protein. And it turns out that this doesn't just bind fatty acids, it also binds siderophores. And in fact, it binds enterobactin. Here's the structure of enterobactin bound to the chicken. So if you see up here is the siderocalin bound to human. This is what we've been looking at all along. And they also have important residues. Here's lysine 125 again. The chicken actually has an arginine on that side, and so you might see that in yours. It still binds enterobactin, but it's making slightly different interactions with it. And the biggest thing about the chicken one, though, is if you look down here, its pocket, you see the 3 pockets for the 3 rings, but it's got this little groove off to the side. And in fact, this little groove has a function, because some bacteria are tricky. They've noticed that all of these mammals are able to steal away their siderophore. And so what they do, they mutate the siderophore. They basically take a glucose molecule, and here's the glucose right here, and they tack it on to the back side of the siderophore. No longer will the siderophore fit into the same pocket, except if the pocket itself mutates to account for that. So the chicken, in this case, has mutated a groove that will bind this sugar over here, and you can see this is its shape is sticking out here at the 9 o'clock position. This is what we call our 9 o'clock groove, and again, this is our common way of looking down at the groove. We set lysine 125 at the 12 o'clock, and we put that means that the binding groove will be at the 9 o'clock if it is there. Notice that chicken has this. Siderocalin does not have as big of a groove, the human one. So that's one of the biggest questions. You've got this glucose, and it fits into this groove in the chicken, and we can know some of the mutations that chicken has that allows that groove to be open enough that a glucose can fit inside it. And that's what you want to look. You want to see if there's a groove. You want to see if you can measure it. And the one thing you've got to remember, proteins can move, especially loops can move. So you've got to look to see there's a loop in the area. In the chicken, the loop is mutated so that it's very short and out of the way. In our other proteins, the question is, can it move out of the way? And you've got to use your imagination and measure it and tell me what you think. So basically we want to know, does your siderocalin look like this with the chicken XFABP? Would it bind, does it have a big 9 o'clock groove? What charge is it? What shape is it? Is it the same, is it big enough to hold a glucose, or is it maybe a little bit smaller? There's a lot of things, and I don't know until you look at it. So here's another view of siderocalin where they clip it from the side. If you can do this, it's a nice way to look at the cup, and you can clip through it, and you can say there's this deep cup, we're cutting through one of the pockets. You can see that it's blue charged because you're cutting through the electrostatic surface. This is one that binds heme, and so it's a much narrower pocket than siderocalins. And so when we look at siderocalin, we see a wider pocket. Maybe somebody will have something that binds something that's much narrower than siderocalin. These could be mutated to bind something different. So you want to look at all these things. You want to see the size and shape and charge of the cup. And in fact, some of these lipocalins can even mutate so they get a catalytic residue that's going to be, that makes it an enzyme. We'll talk about this more maybe when we get into enzymes. For right now, just say we know the mutation that turns it into an enzyme. It's one mutation on the edge of the cup, and it turns it into a cysteine. And these are called PGDS enzymes, but we haven't gotten to enzymes yet, so I'm not going to show you too much about that, except to say there is a loop on these enzymes, and this shows you how the loops can move. If you look over here, this is a side view of the calyx. And here's the loop. They got crystal structures with the loop in the pink conformation where it's sort of bent over, and a loop in the blue conformation where it's sort of bent up. This is the kind of scale of motion we're talking about when we're asking if your loop can move. So you can go back and look at this and think about how it looks like your situation when you compare it to 1DFV. And if you look at this, it's actually the 12 o'clock position is roughly where we set the 12 o'clock position to cysteine 65 if we're looking at the enzyme. But instead we're looking at the binding of siderophore, so we're going to set the lysine 125, or whatever the equivalent residue is in your particular mammalian model. We're going to set that as our 12 o'clock. So that's part one. We're going to be having a human lipocalin. We suspect that it binds multiple bacterial siderophores. That's what we have to start with. 1DFV does that. We want to see how your lipocalin matches and doesn't match it. And so, in fact, we know that it must bind different things because we've seen it bind different things, but I'll tell you more about that in part two.